Today, we are diving into biological membranes, thin, flexible barriers that protect and organize cells. But these membranes do more than just hold everything together. They also control what goes on inside and outside, keeping the cell running smoothly. There are four main components of membranes that we're going to be focusing on for this section. First, the membrane is a bilayer made of lipids and proteins. Second, the membrane is selectively permeable. Next, the membrane is organized but fluid. And last but not least, the membrane is asymmetric. Going back to 1925, Gorder and Grendel proposed the lipid bilayer hypothesis, a groundbreaking idea in cell biology. By analyzing red blood cell membranes, they discovered that the membrane contained twice the amount of lipid needed for a single layer. This led them to suggest that cell membranes are made of a double layer of lipids, with hydrophilic, or water-loving heads, facing outward, and hydrophobic, or water-fearing tails, facing inward. While their model was incomplete, it was missing proteins and membrane asymmetry. It laid the foundation for a modern understanding of cell membranes. With the discoveries from this experiment, we know the lipid bilayer is made of phospholipids, characterized by a phosphate head and two fatty acid tails, whose properties contribute to membrane behavior. Phospholipids are amphipathic, meaning they have a polar head and nonpolar tail. But what truly drives bilayer formation is thermodynamics. According to the free energy equation, it requires less overall energy to hold phospholipids in bilayer formation, making it the energetically favorable conformation. Now let's talk about something crucial: selective permeability. This means membranes only allow certain molecules to pass through. But why? Well, it all comes down to size, charge, and polarity. Small nonpolar molecules like oxygen and carbon dioxide can slip right through. Small polar molecules like water can pass, but not very easily, usually aided with the help of a membrane transport protein, like aquaporins. Large molecules and charged particles need special transport proteins to help them cross. In that way, membranes are just like dams. Just like a dam controls water flow to generate energy, the cell controls ion movement across the membrane to produce power. This is how mitochondria make ATP. Another key concept is membrane fluidity, how much the membrane can move and change shape. Lipids make up the membrane, and their arrangement affects how flexible or stiff it is. If it's too stiff, the cell can't function well. If it's too fluid, things might move out of place. The cell keeps a perfect balance by adjusting the types of lipids and proteins in the membrane. Finally, let's talk about membrane asymmetry. The inside and outside of a membrane are not the same. Different lipids, proteins, and even carbohydrates are arranged on each side to help the cell communicate, transport molecules, and maintain its structure. Special enzymes called flipases and scramblases help arrange these molecules properly. Ultimately, membranes are dynamic, complex, and essential to life. Understanding them helps us learn how cells function and how we can target diseases at the cellular level.
We previously went over the characteristics of a lipid, including the nonpolar lipid tails and the polar head group. Now, what we didn't mention is how this fluid mosaic model, the name given to this bilayer, came from. So let's assume this lipid is starting here. The fluid mosaic model means and encompasses the idea that this lipid actually is moving laterally as well as spinning on its axis. Now, this movement is what allows for permeability because it opens up ways for things to diffuse through and enter in. Now, it's really uncommon is for this lipid to pass through this nonpolar region of lipid tails, because as you see, that polar head group, shown in red, would have to go through this nonpolar region. Hello everybody, and welcome. Today we're going to do an experiment that showcases how the permeability of the membrane is impacted under different temperatures that they're exposed to. So today we're going to be using oil. This is going to be shown in yellow, and that's going to represent your lipid bilayer. Water is going to represent the inside of the cell, and we're going to be using soy sauce to represent any external factors that may be trying to cross the membrane at any point. So why we have two glasses is because this one will be at room temperature, which is going to be our warm environment. This one is slowly getting colder as we're letting the ice dissolve. So these oils are actually the same amount. This ice just needs to chill out for a sec and melt. So let's start with our warm environment. And let's take a look at what happens. Okay, so here goes the soy sauce. We're just gonna gently pour this in there, like this. And we see that it just goes right through the bilayer. So that's kind of that permeability aspect. If we kind of move this around a little bit, there's actually some still stuck in that bilayer. Now at warm temperatures, your lipid membrane is more spread out and it's more fluid, which allows for more things to penetrate. So let's do this again, just to show. Just is able to pass right through that membrane. And if you can see, there's actually still some in that bilayer. But if you do a little shaking around in there, because membranes are fluid, it allows for more to pass through the cell. So this is our warm environment. And we'll cut back when this has officially chilled out. After much waiting, our chilled solution is finally ready to be tested. So here we have again our lipid bilayer and our inside of the cell represented by water. And we are going to add some soy sauce to this now chilled solution. And let's see how fast it goes. Oh, look at that. Now that is cool. So we just saw, like if you remember from this one, how fast this went through. This oil has even changed, and you can see it's very compact because of this decrease in temperature, which is going to decrease the fluidity, increase in compactness, and it's going to limit the permeability of what's going on in there, of that membrane. So if you remember back to the first one, this one was very, very rapid. This one, it took a sec for that soy sauce to dissolve. And that's just showing those different temperatures, how they affect permeability. And other thing I would like to note is cholesterol is something that can be utilized in a membrane, and it can act as a buffer and help in either situation. So if something is very fluid, it can help kind of decrease that fluidity. If something is very cold and compact and less fluid, it can actually help to increase that fluidity of a cell. So thanks for watching. A couple more things to note about how membranes can regulate their fluidity in certain temperatures are the tail length and the kinds of saturated fatty acids there are. So on the left we have the extreme heat condition. In extreme heat we'll see saturated tails more prevalent because those allow for tightly packing, which will help with the high fluidity that arises in warmer conditions. We also see longer tails, as longer tails increase the forces between the lipids, which helps them stay more compact. On the right side, we're going to see more unsaturated tails, which is shown by this kink here, as those are going to help spread out and increase the fluidity in cold temperatures. We also see shorter tails to decrease those interactions in hopes that we can increase fluidity and spread out the membrane more.
Our third topic today is membrane protein structure and function. There are 4 levels of protein structure. Although these levels are increasing in their complexity, folding is not linear, meaning folding does not occur in a sequential order. Rather, folding is determined by the 5 bonding forces at play: covalent bonds, ionic bonds, ion permanent dipole, permanent dipole, and induced dipole. Bonding occurs through thermodynamics that work to increase the stability of the system. The non-covalent bonds occur through backbone-backbone interactions, backbone-side chain interactions, and side chain-side chain interactions. The first structure type we have today is primary structure. This occurs by covalent peptide bonding between amino acid groups. The order of amino acids is important for primary structure. The next structure is secondary. This is a result of backbone interactions through hydrogen bonding. This repeating pattern results in either alpha helices or beta sheets. The third structure type is tertiary, and this occurs when R groups interact with other R groups or with the backbone itself through intermolecular forces such as hydrogen bonds, ionic bonds, ID, and disulfide bonds. Disulfide bonds occur between cysteines to stabilize the structure. These covalent bonds can occur in tertiary structures, and we can see it here where the S is linked to the S. Tertiary structure requires R group interactions with one polypeptide chain, whereas quaternary structure requires R group interactions with more than one polypeptide chain. Otherwise, all of the same intermolecular forces are seen in quaternary structure as are in tertiary structures. Proteins can either exist on the membrane surface or extend into the nonpolar tail region by integrating itself into the membrane. Here we have a different display of proteins in and on the membrane. The first two proteins that we have here are both integral transmembrane proteins that are embedded in the membrane and cross into both the cytosolic and extracellular regions. These proteins are held together by the hydrophobic effect. The third protein we have here in blue is also an integral protein, but it is a monolayer protein, meaning it's only interacting with one layer of the membrane. The last set of proteins we have on the end are going to be an integral protein in gray that is also attached to a peripheral protein on the cytosolic side of the membrane. These proteins are important for providing asymmetry and often bond through hydrogen bonding or intermolecular forces. Now, you may have noticed there are a few different ways to represent proteins through visualization. An important concept is that the structure of the protein dictates its function. We can represent our proteins in a few different ways, but the most common type of visualization is going to be through the ribbon model. This ribbon model can be seen for these alpha helices right here, and then there are a few different displays of primary through quaternary structure with the ribbon model. Our scientist spotlight today is going to be on Jane S. Richardson, who was the creator of the ribbon diagram. Jane Richardson is a pioneering biophysicist whose work can be found in almost every textbook or academic paper that describes a protein. In the 1970s and 80s, the field of structural biology was rapidly expanding. While only around 20 different protein structures had been described, the increasing complexity and similarity of structures made visualization difficult. While wire models were the standard for in-person descriptions, papers often had inconsistent displays of proteins from different viewpoints or perspectives. Richardson spent over half of a year with a pencil and paper trying out different methods and ways to visualize certain proteins. These diagrams were first published in 1981 in the journal Advances in Protein Chemistry in an article titled The Anatomy and Taxonomy of Protein Structure, and since have become a standard for describing protein structure within the field of chemistry. 4 years later, she was awarded the MacArthur Fellowship. Her education background includes a bachelor's degree in philosophy with a minor in physics from Swarthmore College in 1962, followed by graduate work in philosophy at Harvard University. Jane S. Richardson was able to change the way that we see and visualize proteins today.